Hello, class. So we're going to spend some time talking about what happens at the terminal end of a neuron cell, what happens at the synapse. The synapse is the space between the ending of one neuron and the beginning of another. So we call that the presynaptic cleft and the postsynaptic cleft, meaning that you've got your presynaptic, the side that is going to be generating a signal, and then the postsynaptic, the side that is going to be receiving from one cell body to another. As a quick review, you learned in a previous lecture that the action potential is generated by an influx of sodium ions across the cell membrane to depolarize the cell membrane to go from a resting potential of negative 70 millivolts to the threshold potential of negative 50, at which point, once the depolarization hits the threshold, then an action potential will be fired in an all or nothing fashion. So we've learned that the sodium potassium pump is both passive and active depending on what stage you are in the process, but it all is working around this idea of a charge gradient, having pluses on the having more of a negative charge on the inside and a positive charge on the outside. And as that shifts, you're going to get a passive flow of ions, or at the very end, to reestablish that gradient, you're going to have an active transport. Remembering that most of what's going to be talked about in neuronal chemistry is going to be based on this charge gradient. In chemistry, we all know that positives and negatives repel, and so we've got some of that going on, and we also know that the positive will flow towards the negative side and negative will flow towards the positive side. So in addition to the idea of an action potential being propagated, keep in mind that for every single neuron, there are thousands of synapses that are at the terminal end of each of these dendrites. So if you think about that, then you might start to realize that it's more of a democratic sort of conceptual idea, in that you might have many impulses coming from either different places, so a spatial summation where you have impulses coming from several neurons at the same time, reaching the synapses at the same time, or you can have a temporal summation, which is having many signals coming to the same dendrite, or the same synapse from different places over time. Either way, you can have a summation or a cumulative effect, which might actually cause an action potential where an action potential might not have occurred based on just one signal. So this idea of having action potentials generated from multiple signals is very similar to what we're going to talk about when we're talking about neurotransmitters and the idea of cumulative effect. So today we're going to spend a lot of time talking about the synapse. So at a synapse, we're going to specifically talk about the ending of one nerve body or nerve ending and then the beginning of another. So in the previous lecture, you learned a lot about how the axon will be propagated down the cell body, boosting its signal because of myelination. When it gets to the terminal end, this is all the stuff that's going to happen between cells. So just as a quick review, a vesicle is going to be a small little packet. It's a small quantized packet of, quantized meaning that it's going to be a set number of particles. It's going to have a set number of neurotransmitters kind of bundled together. The idea of exocytosis, which is going to happen over here, is the idea of that vesicle bursting. So keep that in mind as I go through this diagram. So the vesicle is going to be synthesized inside the cell body. This vesicle then is stored at the presynaptic cleft, waiting to be released. So what happens when an action potential generates and it comes down this direction, when that action potential generates, it depolarizes the entire cell body. So when that action potential comes down and changes the voltage, the calcium ion, which is also an ion, it's a 2+ charge, the calcium ion will then be initiated to enter into the cell body. So when the calcium ion influx happens, the calcium ion channels open. the neurotransmitters will then be signaled to release their vesicles. So, or the vesicles will be signaled to travel down and release into the synaptic cleft. So, as the action potential causes the calcium to enter, releasing the neurotransmitter, then the neurotransmitters will be let go through exocytosis into the synaptic cleft. Once that neurotransmitter is in the synaptic cleft, it has a variety of options. It can hang out there. It can be picked up again. It's called reuptake when it's taken back up into the cell body. It can be broken down into non-active forms. We'll talk a little bit about acetylcholine. Acetylcholine can be broken down by something called acetylcholinesterase. Whatever the case may be, the duration of time that the neurotransmitter is in this synaptic cleft area is important. So, how long the neurotransmitter stays in there, how many neurotransmitters stay in there, and how many will travel across to the other side, is basically what determines what kind of signal is going to be transmitted. So, when everything sort of works on a positive feedback, positive negative feedback loop. So, you might have something. You might have the neurotransmitters released, and then the signal will say, okay, that's enough. And so something else, the postsynaptic cell might release retrograde transmitters that, as you can see here, slow further release of the presynaptic cell. There's communication between the presynaptic cleft and the postsynaptic cleft. This is post and the pre. So that not so much of the neurotransmitter just hangs out there for too long. Okay, so chemical events at the synapse. So there are some major categories of neurotransmitters. Some of them are going to be amino acids. So if you remember from biology, amino acids are the basic building blocks of DNA. So these are kind of all over the place, right? So we have glutamate and GABA. We have, you might hear those terms. We have acetylcholine, which is, I think, the first neurotransmitter to be discovered or to at least be understood. We have certain things called monoamines, which are amines or NH compounds, nitrogen ammonia or nitrogen hydrogen compounds. Those are serotonin, dopamine, norepinephrine, epinephrine. You might have heard like EpiPen before. Norepinephrine and epinephrine are what you think of when you think of adrenaline. And purines, adenosine, and gases such as nitric oxide, not laughing gas, which is nitrous oxide, that are oftentimes used that can cause different effects when they are used as neurotransmitters. We are going to talk a lot about some of these neurotransmitters, and some of them will be discussed throughout the course. Some of them won't be discussed at all, but for the most part, they all fall into two different categories of inhibitory and excitatory. And just as you can imagine, inhibitory and excitatory means exactly what they sound like. Inhibitory neurotransmitters will inhibit a signal from transferring from cell to cell, and an excitatory neurotransmitter will be a green light. It will excite more action potentials so that you will have more of a physical response. Inhibitory responses might just be telling the cell body to actually stop sending the signal. For instance, slowing your heart rate, increasing your blood volume, so slowing your heart rate. And then an excitatory signal might be, this is actually easier to think about. Excitatory would be something like muscle contractions, having more blood flow, things like that. The excitatory is probably what you're more familiar with when you think of like norepinephrine and epinephrine, getting excited. Dopamine is in the category of neurotransmitter. They can do both. And serotonin is in the category of well-being, which is kind of a calming. So different neurotransmitters have different functions, and we'll talk a little bit more about that as it becomes applicable to our class. what a neurotransmitter can do once it gets to the synaptic cleft and it gets to the binding site, which is on the other side. When it binds to the receptor site on the postsynaptic cleft, it can have an ionotropic effect, which is it can actually affect the ion gradient. So it can open ion channels, increasing the sodium potassium pump, which means you can have a lot more action potentials. Excitatory neurotransmitters can oftentimes have this effect. Acetylcholine, dopamine, things like that. Metabotropic effects can be a longer lasting effect when a neurotransmitter attaches to a receptor and it initiates a sequence of events. For instance, when the neurotransmitter signals to the neuron to release hormones, for instance, that might actually initiate a sequence of metabolic reactions that are slower but go on for longer. Antagonist drugs. So, one of the ways that we know a lot about neurotransmitters is by trying to stabilize when there's a neurotransmitter imbalance. So, when it's medication or illegal drugs, also have this effect of when they interrupt the natural balance of neurotransmitters, we learn a lot about how neurotransmitters work. Basically, by messing with them, we've learned a lot about them. An antagonist drug blocks the effect of a neurotransmitter, meaning like an antagonist in a play or something. It actually it blocks the effect of the normal neurotransmitters getting to where they need to dock. So Novocaine and caffeine. Novocaine, obviously, for pain, blocks pain receptors, and caffeine to keep you stimulated. An agonist drug might mimic or increase the effect of the neurotransmitters. So something like LSD or cocaine, heroin, those are what you think of when you think of euphoric or hallucinogenic, or just being high. And those drugs actually increase the amount of dopamine in the blood system, in the synaptic cleft, either by stimulating a release of a lot of dopamine at once, or by, on the other end at the postsynaptic cleft, opening lots of channels all at once, or actually allowing for the dopamine to stay in the synaptic cleft for a long time. So drugs have only a certain amount of things that, like certain mechanisms by which they can work. They will work either by increasing the synthesis, so increasing the amount of neurotransmitter that's actually produced. They can cause those vesicles, those little tiny bundles, to leak. They can increase the release, like a quick burst of cocaine is really like one of the things that we know a lot about in quick bursts of dopamine release, meaning that you get a very quick high, but then you also get a very quick low upon once the euphoria kind of wears off. You can decrease the reuptake. So something like serotonin, which is a neurotransmitter that's linked directly to the feeling of well-being. When you have deep depression or anxiety, a lot of times serotonin is the neurotransmitter that we try to balance. And serotonin SSRIs, if you've heard of those, is a drug category that helps with serotonin reuptake, making serotonin reuptake take longer, which allows for the serotonin to remain in the synaptic cleft a little bit longer. So if you decrease the reuptake, it kind of has that opposite effect of having a longer lasting feeling of wellness, for serotonin at least. Blocking the breakdown into inactive chemicals. So acetylcholine is an excitatory neurotransmitter that oftentimes is linked towards muscle contractions and things like that. If you want those muscle contractions to last longer, or if you want to control those, blocking the breakdown of that neurotransmitter is probably the way to go. And then finally, directly stimulating or blocking postsynaptic receptors. This is one of the main reasons for, if you think about blocking the place where the neurotransmitter has to dock, then it's the idea that it doesn't really matter if you have the neurotransmitters releasing. You can actually block the neurotransmitters from being read or understood. So finally, when a medication or a drug has an affinity for a particular type of receptor, that's one thing. But that idea of binding to the site is also very important. So as we talk further about treatments and medications, the idea that you have to have both efficacy and affinity, it becomes really important. So it's this balance between finding a way to create a synthetic version of a neurotransmitter that can have the same affinity for the receptor, and it can have the efficacy that it needs to trigger the receptor site to signal to the cell, to the nerve cell, that it needs to do something. Both of these factors are very important, and so that balance between having something synthetic do what a neurotransmitter can do is the conundrum, is what we try to balance out. So anytime we talk about mental health, a lot of times that the medication oftentimes has to be tweaked so that you can have this balance between making enough and having enough transmitting the right signals. So hopefully you learned a little bit about, in this quick soundbite, of what a synapse is, just the basics of how a neurotransmitter works, why the neurotransmitters are important. We'll delve a little bit deeper into this as we become more invested in the course, and you might, especially during the addiction unit, will spend a lot of time talking about dopamine and the reward pathway in the brain, which is always really interesting. And then when we talk about Parkinson's and Huntington's, we'll talk a lot about acetylcholine. So hopefully this is a quick beginning, and you had a good chance to at least be introduced to synapses.